My name is Junker Naranjo, and in this conference, while talking about glycolysis, gluconeogenesis, and glycogen metabolism. In first place, we will talking about glycolysis. Glycolysis is the main role for glucose metabolism and the main role for metabolism of fructose, galactose, and other carbohydrates derived from the diet. The ability of the glucose to provide ATP in the absence of oxygen is especially important because this allows the shredded muscle to perform at very high low operating cost when the oxygen supply is insufficient, and it allows the tissue to survive episodes of anoxia. Glycolysis this is a metabolic pathway that converts glucose into pyruvate and energy in the form of ATP. The following are steps of glycolysis. 1 steps. Glucose phosphorylation. Glucose is phosphorylated by the enzyme hexokinase using a molecule of ATP to form glucose 6-phosphate. 2 steps. Isomerization. Glucose 6-phosphate is isomerized to form fructose 6-phosphate, which is catalyzed by the enzyme phosphoxylase isomerase. 3 steps. Second, phosphorylation. Fructose 6-phosphate is phosphorylated by the enzyme phosphofructokinase 1 using another molecule of ATP to form fructose 1,6-biphosphate. 4 steps. Cleavage. Fructose 1,6-biphosphate is cleaved into 2 3-carbon molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by the enzyme aldolase. 5 steps. Isomerization. Dihydroxyacetone phosphate is isomerized into glyceraldehyde 3-phosphate by the enzyme triose-phosphate isomerase. 6 steps. Oxidation and phosphorylation. Glyceraldehyde 3-phosphate is oxidized by the action of the enzyme glyceraldehyde 3-phosphate dehydrogenase, generation NAD and molecule of 1,3-biphosphoglycerate. This molecule is then phosphorylated by the enzyme phosphoglycerate kinase, using a molecule of ADP to form ATP and molecule of 3-phosphoglycerate. 7 steps. Rearrangement. 3-phosphoglycerate is rearranged into 2-phosphoglycerate by the action of the enzyme phosphoglycerate mutase. 8 steps. Dehydration. 2-phosphoglycerate is dehydrated to form phosphoenol pyruvate by the action of the enzyme enolase. 9 steps. Final phosphorylation. Phosphoenol pyruvate is then phosphorylated by the enzyme pyruvate kinase using a molecule of ADP to form ATP and pyruvate. Overall, glycolysis is a process a net of 2 molecules of ATP and 2 molecules of NAD from 1 molecule of glucose. The pyruvate process in this process can then enter the citric acid cycle to further energy production. Although most of the glycolysis reactions are irreversible, 3 are largely exergonic and 3-4 should be considered irreversible from the physiological point of view. This reaction catalyzed by the exokinase and glucosinase and phosphotosinase and the pyruvatokinase are the main sites of regulation of glycolysis. Okay, now we will talking about gluconeogenesis. Gluconeogenesis is the metabolic pathway in which the body produces glucose from non-carbohydrate such as amino acids, lactate, and glycerol. This process occurs primarily in the liver and is an important part of the regulation of glucose homeostasis. Gluconeogenesis is essential for maintaining blood glucose level during periods of low carbohydrate availability such as fasting or exercise. The primary enzymes involved in gluconeogenesis are different from those involved in glycolysis. The process by which glucose is broken down to produce energy. This ensures that the two processes can be regulated separately, allowing the body to maintain balance between glucose production and utilization. Sure, here are all steps of gluconeogenesis. 1 steps. Pyruvate carboxylation. Pyruvate is carboxylated to form oxaloacetate with the enzyme pyruvate carboxylase. 2 steps. Oxaloacetate reduction. Oxaloacetate is reduced to malate by the enzyme malate dehydrogenase. 3 steps. Malate transport. Malate is transported from the mitochondria to the cytosol. 4 steps. Malate oxidation. Malate is oxidized to oxaloacetate by the enzyme malic enzyme, producing NADPH. 5 steps. Oxaloacetate decarboxylation. Oxaloacetate is decarboxylated to form phosphoenol pyruvate with the enzyme phosphoenol pyruvate carboxygenase. 6 steps. Fructose 1,6-biphosphate. Fructose 1,6-biphosphate catalyzes the hydrolysis of fructose 1,6-biphosphate to form fructose 6-phosphate and inorganic phosphate. 7 steps. Glucose 6-phosphate. Glucose 6-phosphate catalyzes the hydrolysis of glucose 6-phosphate to form glucose and inorganic phosphate. 8 steps. Glucose transport. Glucose is transported out of the cell and into the blood stream. These steps are used to synthesize glucose from non-carbohydrate sources, such as amino acids, lactate, or glycerol, when glucose is needed but not available from the diet. It is important to note that gluconeogenesis is not simple reversal of glycolysis, as certain steps in the process are unique to gluconeogenesis and are not found in glycolysis. Additionally, some of the enzymes involved in gluconeogenesis have a different regulation than their counterparts in glycolysis, allowing for the separate regulation of the two processes. The metabolism of glycogen refers to processes by which glycogen is synthesized and degraded in the body. Glycogen synthesis, also known as glycogenesis, is the process by which glucose molecules are linked together to form glycogen. This process occurs primarily in the liver and muscle cells and is stimulated by insulin. During glycogenesis, glucose is converted to glucose 6-phosphate by the enzyme hexokinase. Glucose 6-phosphate is the convert to glucose 1-phosphate and 10 to UDP glucose, which is used to form glycogen. The enzymes involved in glycogen synthesis include glycogen synthase, UDP, glucose pyrophosphorylase, and glycogen branching enzymes. Glycogen degradation. Glycogen degradation, also known as glycogenolysis, is a process by which glycogen is broken down into glucose molecules. The process occurs in response to the hormone glucagon and occurs primarily in the liver. During glycogenolysis, glycogen is broken down into glucose 1-phosphate by the enzyme glycogen phosphorylase. Glucose 1,6-phosphate is converted to glucose 6-phosphate, which can be used as an energy source. The balance between glycogen synthesis and degradation is tightly regulated by hormones such as insulin and glucagon. Insulin stimulates glycogen synthesis and inhibits glycogen degradation, while glucagon stimulates glycogen degradation and inhibits glycogen synthesis. This allows the body to quickly respond to change in glucose demands and maintain a stable blood glucose level.